Our team is pleased to present a research proposal on investigating sublethal effects of imidacloprid exposure on little brown bats in central Canada. With the ability to eat close to their entire body weight in insects each day, bats play a crucial role as biological pest controllers. In recent years, losses of bats have reached well into the millions, causing concern for the eventual regional extinction of entire colonies, including those of the little brown bats. It is believed that long-term exposure of insecticides on bats may cause immunosuppression, leaving colonies vulnerable to issues including white nose syndrome. This is an integral example of an endangered biocontrol system whose welfare is underrepresented in current research studies, and for this reason it forms the basis of the following research proposal.
Little brown bats are listed as endangered species by SARA, the Canadian Species at Risk Act. Populations were severely depleted after the outbreak of white nose syndrome, which spread rapidly throughout hibernating colonies. Between 2006 and 2012, an estimated 5.7 to 6.7 million bats were lost due to this fungal disease. White nose syndrome came to attention in the same year beekeepers realized imidacloprid was a contributing factor to colony collapse disorder. This raised concerns over the impact of insecticides on bats. As one of the most effective biocontrol agents, the agriculture industry should have an invested interest in preserving and fostering little brown bat recovery. And this can start by figuring out whether current use of neonicotinoids is inhibiting population recovery. What we do know is that while neonicotinoids aren't generally thought of as toxic to mammals, adverse impacts have been documented. And research suggests that long-term exposure to neonicotinoids leaves bats with a weakened immune system and thus vulnerable to diseases. We also know that little brown bats are likely to be exposed to neonicotinoids and due to their physiology are likely to experience toxic effects. These bats are highly active in agricultural systems and are avid consumers of herbivorous insects. Also, bats use finely adapted vocal motor systems to forage and navigate. Many parallels can be drawn between their sensory system and that of bees, a species that has been thoroughly documented as a victim of neonicotinoid use. Moreover, these small mammals have a long lifespan, up to 30 years, and could be accumulating chemical residues or passing residues to their young via lactation. Despite this, very little is known about the specific effects of neonicotinoids on bat behavior and health. Effect concentration and what those effects would be still remains unknown.
The purpose of our study is to gain a better understanding of the adverse effects that neonicotinoid usage has on little brown bats. Neonicotinoid usage is prevalent within agriculture settings due to its ability to control a variety of pests on a wide scale. Unfortunately, since these pesticides do not have a specific pest they target, they ultimately have a negative impact on other species such as the little brown bat. We will be conducting our research by utilizing the exposure rates of imidacloprid and its sublethal impacts that the pesticide will have on the species. Since neonicotinoids target the nervous system of pests, we will be studying the foraging behavior of the bats to clearly determine whether their foraging patterns have been altered. The study will be conducted over the duration of 5 years, which will allow adequate data to be collected on the degree of impact on the species. The duration of the experiment will allow us to determine if longer exposure rates have a more negative impact on the species. We hope to determine insecticide application benchmarks that can be safely administered without having negative impacts on bats. Our research will be required to fill the knowledge gaps of effects which neonicotinoids such as imidacloprid may have on species, like the little brown bat. These species are vital within ecosystems, and if negative impacts are measured in the experiment, it will ultimately mean that the pesticide has an overall impact on the ecosystem. These species are prominent within agriculture settings for their efficiency in arthropod management. This research will benefit the agricultural setting along with the species themselves.
Study design. 40 little brown bats will be collected from the same colony near Kearney, Ontario. The bats' legs will be banded with an individual ID number and collected with a government permit. The bats will be divided into 4 groups of 10 and housed in group-specific artificial outdoor roosts. Treatment groups 1 and 2 will be exposed to low and high levels, respectively, of imidacloprid through contaminated prey. To develop these contaminated prey populations, herbivorous imidacloprid-resistant or tolerant organisms will be obtained from the Harrow Research and Development Centre in Harrow, Ontario. Prey for groups 1 and 2 will then be reared in simulated crop environments. Twice per year, they will receive either low or high rate foliar applications of Admire 240 flowable systemic insecticide, once at the traditional economic threshold point and once 2 weeks later, mimicking intended usage. The low-rate conventional crop environment will receive 100 milliliters per hectare treatment of Admire, and the high-rate conventional crop environment will receive Admire at 200 milliliter per hectare treatment. Treatment Group 3 will be exposed to prey having been raised in a similar simulated crop environment, but it will receive a single in-furrow soil application of Admire at 10 milliliters per 100 meters of row. No other synthetic application will be used. Treatment Group 4 will be a control. Bats in this group will be fed from a prey population reared in a simulated organic crop environment with no exposure to imidacloprid. To summarize, bats in Group 1 will be introduced to prey exposed to a low rate of imidacloprid biannually. Bats in Group 2 will be introduced to prey exposed to a high rate of imidacloprid biannually. Bats in Group 3 will be introduced to prey exposed to a single in-furrow application of imidacloprid. And finally, bats in Group 4 will be a control group introduced to prey with no exposure to synthetic applications.
During the 5-year experiment period, negative influences from imidacloprid can be shown through observing any changes in call behavior. This can provide an estimation of the effect concentration of imidacloprid. With the effect concentration known, both hazard quotient and margin of safety can then be calculated in other ecosystems of interest by taking their highest exposure concentration of imidacloprid. The hazard quotient can be calculated by dividing the exposure concentration by the effect concentration. The margin of safety can be calculated by dividing the effect concentration by the exposure concentration. A range of effect concentration values from different ecosystems can be used to calculate a probabilistic risk assessment by comparing the toxicity distribution curve to an exposure distribution curve. The probabilistic risk assessment value is a crucial component in establishing guidelines for pesticide regulation and management according to the Pest Management Regulatory Agency, which integrates a variety of risk and value assessments to provide the Canadian government with proper regulation and management on pesticide use, such as imidacloprid.
The data that this study proposes to gather regarding imidacloprid's sublethal effects, including those on vocalizations, restful sleep, and other physical attributes of little brown bats, will help us develop nature's scorecard on the health impacts of this unplanned exposure to neonicotinoids. The alarming rate at which the bat population is depleting is a call to action that cannot be ignored, as they provide billions of dollars of pest control for agriculture each year. With this proposal, the intent is to fill the most apparent knowledge gaps and foster a principal understanding of the effects of neonicotinoids on the bat population.